Hello everyone, today we are here to discuss about Biopharmaceutics and Pharmacokinetics previous year questions, which is presented by Vittal Patil, Pritam Patil, Anuj Bhosle, Yash Sadhav and Rushikesh Mali. We are the student of Ashok Ramani Institute of Pharmaceutical Sciences and Research, Save. This video is guided by Mr. A.N. Dhage sir. In this video, we discuss about the questions. The question 1 is explain the methods of measurement of bioavailability, and question 2 is explain two-compartment open model for the IV bolus administration. Question 1. Explain the methods of measurement of bioavailability. Definition. Bioavailability is defined as the rate and extent of absorption of unchanged drug from its dosage form and becomes available at the site of action. Measurement of bioavailability. A. Pharmacokinetic method. In this, plasma level time studies, urinary excretion studies. B. Pharmacodynamic methods, acute pharmacological response and therapeutic response. Pharmacokinetic method. First, plasma level time studies. Principle. The method is based on the assumption that two dosage forms that exhibit superimposable plasma level time profile in a group of subjects should result in identical therapeutic activity. These studies can be single dosage or multi-dosage studies. Second, urinary excretion studies. Collection of urine at the regular interval for a time span equal to the 7 biological half-lives. Analysis of unchanged drug in the collected sample. Determination of amount of a drug excreted in each interval. Note, at each sample collection, total emptying of a bladder is necessary to avoid errors resulting from the addition of residual amount to the next urine. Second, pharmacodynamic method. First, acute pharmacological response. It may be include the measurement of one or more following pharmacological response. Change in ECG or EEG reading, pupil diameter, etc. are the related to the time course of the given drug. Disadvantages. First, variable pharmacological responses. Second, accurate correlation between the measurement response and the drug available is difficult. Therapeutic response principle. Observing the clinical response to the drug formulation given to the patient suffering from the disease for which it is intended to be used, but the method is associated with the fact that the response observed is often too improper. Pharmacodynamic studies are not recommended for the oral administration of the drug product. Question 2. Explain two-compartment open model for IV bolus administration. Two-compartment open model. Compartment. A compartment is not a real physiological or anatomical region but an imaginary or hypothetically consists of group of tissue. In this model, the drug distributed into the two compartments. It is central compartment and peripheral compartment. Central compartment or compartment first. In this compartment, comparison of blood and highly perfused tissue like liver, lungs, etc. Peripheral or tissue or compartment second. In this compartment, comparison of poorly perfused and slow equilibrating tissue such as muscle, skin, adipose tissues, etc. There are several possible two-compartment models. Model A, Model B, Model C shows in a figure. Two-compartment open model intravenous bolus administration. The model can be shows in a figure. In figure, let's K12 and K21 the first order distribution rate constants shows drug transfer between the central and the peripheral compartment. The rate of change in the drug concentration in the central compartment is given by dC upon dt is equal to K21 Cp minus K12 Cc minus Ke Cc. Extending the relationship x is equal to Vd into C to the above equation, we have dCc upon dt is equal to K21 into Xp upon Vp minus K12 into Xc upon Vc minus Ke into Xc upon Vc, where Xc and Kp are the amount of drug in the central and peripheral compartment respectively. Vp and Xp are the apparent volume of the central and peripheral compartment is given by dCp upon dt is equal to K12 into Cc minus K21 into Cp is equal to K12 Xe upon Ve minus Kxp upon Vp. Integration of equation 2 and 4 yields the equation that describe the concentration of a drug in the central and peripheral compartment at any given time t. Cc is equal to Xo upon Vc in bracket K21 minus alpha upon beta minus alpha into e raised to minus alpha t plus K21 minus beta upon alpha minus beta into e raised to minus beta t. Cp is equal to Xo upon Vp in bracket K12 upon beta minus alpha into e raised to minus alpha t plus K12 upon alpha minus beta e raised to minus beta t, where Xo is the IV bolus dose, where alpha and beta are the hybrid constant, and K12 and K21 called as a micro constant, and their relationship is given as alpha plus beta is equal to K12 plus K21 plus Ke. Alpha into beta is equal to K21 into Ke. Equation 5 can be written in the simple form as Cc is equal to A into e raised to minus alpha t minus b into e raised to minus beta t. Method of residual. The bioexponential equation 9 can be resolved in individual components by the method of residual. When the distribution is more rapid than the elimination, the rate constant is alpha is greater than the beta. The term e raised to alpha t approaches to the 0 and the equation reduces to the C is equal to b into e raised to minus beta t. In log form, the log C is equal to log b minus beta t upon 2.303, where C is the back extrapolated plasma concentration value and a semi-log plot of the C versus St yields terminal linear phase of the curve having slope minus beta upon 2.303, and T half or elimination phase is given as a T half is equal to 0.693 upon beta. Subtraction of extrapolated plasma concentration value from the corresponding through the plasma concentration value. A series of residual concentrations is equal to Cr is equal to C minus C is equal to A into e raised to minus alpha. In log form, log Cr is equal to log A minus alpha t upon 2.303, a number of pharmacokinetic parameters at t is equal to 0. The equation 9 reduces to C0 is equal to A plus B, Ke is equal to alpha plus beta in bracket A plus B upon A into beta plus B alpha. K12 is equal to A into B in bracket beta minus alpha upon in bracket A plus B into in bracket A into beta plus B alpha. K21 is equal to A beta plus B alpha upon A plus B.